Okay, so what you have then with these myofilaments is there's two kinds that are sliding alongside each other, and that's sort of the essence at the microscopic level of muscle movement. So what you have is the thick filaments and the thin filaments. The thick filaments are composed of a single protein, myosin, several different protein molecules in each myofilament. And myosin is this very interesting structure where you see this line, this tail of myosin attached to a myosin head. And the myosin head is the thing that's actually going to be interacting with the other type of myofilament, the thin filament. Now the thin filament is composed of primarily of three different proteins. There's lots of others too. These are actin, troponin, and tropomyosin. Now the deal is how this is organized is in this interesting way. The actin molecules form these globular molecules which form this sort of helix, kind of like what you see with DNA. Okay, these sort of two strands sort of winding around each other. All right. Then you have these troponin molecules, these little dark brown globs here in this illustration, and tropomyosin, these sort of stranded molecules running along kind of the grooves of this chain of actin molecules. Okay. And so what's going to happen then, again, these things are going to be sliding across each other. Okay, and that's what we refer to as this idea of sliding filament theory. Now, how does this sliding happen? Okay, what you've got to start off with is the fact that this muscle has to be activated for it to slide. Or well, there's other ways to do it, but we'll talk about that in a second. But let's just talk about a muscle shortening, and in the case where there's some force being exerted on it that you need to overcome. Okay, so we want to lift, we want to shorten our bicep to lift a weight. Okay, or something like that. So what's going to happen then is that neurons in our brain and in our spinal cord, well, ultimately in our spinal cord, will project down into the muscle and actually make a contact with a particular synapse. Will make a contact with an individual muscle fiber. Okay, this is what I think is kind of cool. For every muscle fiber, there's going to be some kind of neural connection. Now, a particular neuron in the spinal cord can connect to many muscle fibers. And again, this is something we won't talk about in this class, but we'll talk about in Bio 45100. In any case, what will happen then is that this neuron will generate at some point by command of the brain, or even due to reflexes or some other sorts of neural activity in the spinal cord, will generate action potentials. These action potentials will travel down this neuron to the synapse, and it will depolarize the muscle membrane, just like you can have synaptic transmission that can have an effect on a postsynaptic cell. In this case, in this neuromuscular junction, you have a synapse on the muscle fiber itself. Okay, so then this depolarization will travel along the muscle and actually travel into the muscle fibers, to a point where it's adjacent to all these different myofibrils that you're seeing here. Okay. Now, what happens then is that membrane in a particular organelle within the muscle cell called sarcoplasmic reticulum will depolarize. I'm going to go over the rest of these steps, and then we'll go into them in a little quickly, and then we'll go into them in a little bit more detail. That's going to open up calcium channels in the sarcoplasmic reticulum, and that's going to allow then calcium to be released from the sarcoplasmic reticulum into the sarcoplasm. What the sarcoplasm is basically just the interior. Let's just think of it as a cytoplasm, but applied now for muscle cells, okay, for muscle fibers. So a sarcoplasm is basically just the interior of a muscle cell. Okay, then calcium is going to bind to troponin, and what that's going to do then, we'll see this in a second, is it's going to allow these thin and thick filaments to now connect together and begin to crawl across each other, and thus change the length of the muscle. Okay, so here's just zooming in, sort of a cartoon of what's going on in terms of this activation. So you have this potential, this membrane potential, traveling into the interior of the muscle fiber. Okay, it becomes a location adjacent to the sarcoplasmic reticulum, and what the sarcoplasmic reticulum is basically this organelle, a little bit like endoplasmic reticulum, but specialized specifically for muscle cells. what it's doing is it's got ATP pumps that are pumping calcium in constantly and keeping the calcium concentration very high inside of it relative to the outside inside the sarcoplasm. Now, when depolarization occurs, this will open up gates within the sarcoplasmic reticulum, and now basically it's just increasing calcium permeability. So now calcium can flow down its concentration gradient from the interior of the sarcoplasmic reticulum into the sarcoplasm. And when this calcium concentration increases, that's going to increase the possibility that calcium will bind to this molecule troponin, which, as I mentioned, is part of this thin filament. It's basically kind of a regulatory protein in terms of myofibril function. Now, when that happens, this is ultimately going to allow these actin molecules and myosin molecules, actin molecules in the thin filaments and myosin molecules, which again this composes the thick filament, to connect. Okay, and once these guys can connect, they can begin to start moving. Okay, and this is referred to as the power stroke here in step 7. We're going to look more into that right now. Okay, so what you're seeing here is the thick filament. In this cartoon, you got a thick filament, okay, with a myosin head, the thin filament here. And now what's happened is, because you've got this binding with troponin, is that these myosin molecules and actin molecules can begin to interact. Now what you also need here is the presence of ATP, okay? And what's going to happen is that at the start here is that actually an ADP molecule, the thing that basically can create an ATP with an addition of a phosphate, is attached to this myosin head. It will, once this connection happens, it will actually fall off and basically allow this movement of the myosin head from this original position like you're seeing here, basically changing position, sort of doing this in this diagram, at least like a counterclockwise rotation. And this will basically then act to move this actin molecule, in this case to the left, relative to the thick filament. So the thin filament will move to the left with respect to the thick filament. Okay, so again, it attaches to the actin and then rotates, and this allows this movement to occur. Okay. Then what will happen is that ATP will reattach to this myosin molecule, and actually where you need the hydrolysis of ATP, basically yielding the energy of ATP, is actually not to move that muscle, but ultimately to basically recock it. So what will happen here is that ATP will attach to the myosin head. That will actually allow the myosin head to detach from the thin filament. Okay? This then, once this detachment has happened, now the hydrolysis of ATP will actually recock this myosin head so it goes back into its original position, like you see here. Okay? And once that happens, then this cycle can start over again, because if there's still calcium present, once it's detached but recocked, it can now reattach basically to another actin molecule, and this cycle will keep repeating. So basically, attaching, rotating, dragging the thin filament relative to the thick filament, detaching with the presence of ATP, hydrolysis of ATP, allowing it to recock, and then repeating. Okay, and this is what was referred to as the cross-bridge cycle. And now, at a microscopic basis, this is ultimately how, mechanistically now, how thin and thick filaments are moving relative to each other, and basically, in this case, allowing muscle to shorten. Okay, so some of the things I want to talk about. So in this particular case, this where the thin filament is moving relative to the thick filament due to this cross-bridge cycle, this is actually referred to as what's referred to as a concentric contraction. That's what you're seeing here. In this case, in this illustration here, it's actually your deltoid muscles in your shoulder that are acting to basically move your entire arm. For a bicep, it's basically for this flexion to occur. Okay, for that elbow joint angle to get smaller. So what's happening here is that you have the muscle being activated, okay, and this is actually referred to in muscle physiology parlance as contraction. Okay, contraction in muscle parlance is the activation of the muscle itself. Okay, the ability now for these thin and thick filaments to interact and start walking across each other. And if in the case of this elbow joint shortening, it's referred to as a concentric contraction. Okay. However, this isn't the only possibility of what happens during a muscle contraction. Okay. We know, for example, that if you're just going to be holding a book, let's say, and you'll be doing this basically in the lab, the equivalent with this arm model. Let's say you're just holding a book, okay, but your elbow joint is not moving. Your elbow joint, the angle is not changing. Basically, your arm is still. And yet, of course, you're exerting force on this book. And if you didn't exert the force, the book would fall down, right? So what's happening there is what's referred to as an isometric contraction. So what's happening is that the muscle is being activated, but yet the joint is not actually moving. Now you can actually go further, going even beyond that to what we refer to as an eccentric contraction. What's happening there is that it's an example that would be something like you're lowering this book down gently to the ground. Okay, if you didn't have any muscle activation at all, it would just plop down on the ground, just following gravity, dictated by gravity. But using your muscle, what you're doing is you're gently lowering it to the ground. Okay, you're exerting force. But now your elbow joint, instead of shortening, is actually instead of your elbow joint angle decreasing or producing a flexion. In this case, you're producing an extension. So this is a little bit weird. This is kind of the opposite of what I was showing you at the start of this when I was talking about the different jargon and agonist and antagonist. In this case, the biceps is being activated. Okay, or it's contracting, and yet it's actually an extension is occurring rather than a flexion. And this is referred to as an eccentric contraction. That's what you're seeing here. Okay, now what's going on there? How can you have activation of muscle, but the muscle itself actually lengthening? Well, the explanation is it's actually not too complicated. The deal is that, as I mentioned, you've got this. I'm closing in on one of the steps of the cross-bridge cycle, and this is the step again where ADP falls off, and this yank occurs where this myosin head rotates and tries to pull the actin. In what you're seeing in the cartoon, the actin or the thin filament to the left relative to the thick filament. But imagine, and I don't have the arrow here. Whoops! But imagine you have an opposing force. Now that could be gravity. That could be even the activation of the antagonist muscle, for the biceps, in this case, the triceps. Okay? And if that force, that opposing force, is bigger than the force that the muscle is trying to make, you're actually going to have movement in the opposite direction. Okay? In that case, if you've got the opposing force that's stronger than the force that the muscle is trying to make, you're actually going to have the muscle lengthening. Okay? So, and it's not just quite even that. It's like, and also, once this movement is occurring, we know that actually force is not proportional to velocity. It's actually proportional to acceleration. But the bottom line is that once these opposing forces exceed the force produced by the biceps, and once that relationship is sort of maintained, that relative net force, basically what's going to happen then is that the muscle is going to begin to lengthen. Okay? And what's actually happening there is, I'm not going to show it here, but you're actually getting the thin filaments and thick filaments dragged across each other. Okay? So here's like a cartoon showing that. So in this case, these thin filaments are actually being dragged across the thick filaments. So now in this case, the thin filaments here are moving to the right, even though the thick filaments are attached. And it's a little bit like, I don't want to sound like I'm mean to cats. I actually have a couple cats, but imagine you took a cat, grabbed it by its hind legs, and tried to drag it across a carpet. Okay, it's going to try to pull onto that carpet, but what would happen then, at least if the carpet's attached before, is that it'll just drag itself, across the carpet. So it kind of its claws detach and reattach and detach and reattach. Okay. In this case, what's happening here is the same thing. The thin filament is being dragged across to the right and kind of attaching and detaching, attaching and reattaching to the thin filament. What's interesting here is that you do need calcium for this to occur. You do need the presence of calcium, so you do need this nervous system activation, but you actually don't need any ATP for this to happen. There's some interesting consequences of this that, again, we'll not explore in this class, but if you're interested, you'd see that in, again, Bio 45100. Okay, so I think, that's it. And now I hope you're more ready for the lab, and I hope you now can find it a little bit more interesting than you otherwise would. Thank you so much. Bye-bye.